Hello viewers, you are welcome to my YouTube channel Fasihuddin. In this video, I will be discussing on the development of the embryo sac or the female gametophyte with respect to angiosperms. In the fascinating world of the flowering plants, sexual reproduction relies on the interplay of male and female gametophytes. The female gametophyte, more commonly known as the embryo sac, is a microscopic but crucial structure nested within the ovule of flower's ovary. It represents haploid female sexual phase of the plant's life cycle and is the site of fertilization and the beginning of the seed development. This tiny structure plays a pivotal role in ensuring the successful propagation of the flowering plants. The embryo sac not only merely a passive container, it is a highly organized entity with specialized cells that perform distinct functions. It houses the egg cell, the actual female gamete, which will fuse with the sperm cell during fertilization. Furthermore, it contains supporting cells, the Synergids, which play a crucial role in attracting the pollen tube and facilitating the delivery of the sperm cells. The central cell, another key component, contains the polar nuclei that upon fertilization give rise to the endosperm, a nutritive tissue that will nourish the developing embryo. The embryo sac is essential bridge between the generations, providing the environment and resources necessary for the formation of the next generation of flowering plants. Here are some of the key stages involved in the embryo sac development. The embryo sac develops within the ovule, which is located inside the ovary of the flower. It originates from the megaspore mother cell, abbreviated as the MMC. The birth of the megaspore is known as the Megasporogenesis. It is the process of the formation of megaspores from the megaspore mother cell. This process occurs within the ovule, a part of the female reproductive structure of a flowering plant. The MMC undergoes meiosis to produce four haploid megaspores. Typically, only one of these megaspores survives while the other degenerates. The Megagametogenesis, the surviving megaspore undergoes a series of mitotic divisions and cellular differentiation to form a mature embryo sac. There are different types of embryo sac development in angiosperms, classified based on the number of megaspores that contribute to its formation. For example, monosporic, where one megaspore is involved; bisporic, two megaspores are involved; and tetrasporic, where all the four megaspores are involved in embryo sac development. Embryo sac typically consists of 7 cells but contains 8 nuclei. Egg apparatus comprising egg cell and two synergids, central cell containing two polar nuclei, and antipodals are 3 in number. Monosporic development means the embryo sac or the female gametophyte in the flowering plants originates from the single megaspore. This is the most common type found in the angiosperm. Polygonum is the most classical example, and here is how it unfolds. All it begins with the diploid cell within the ovule called megaspore mother cell, abbreviated as the MMC. It undergoes meiosis, a type of cell division that reduces the chromosome number by half. This results in four haploid megaspores, usually arranged in a linear tetrad. The functional megaspore. Typically, only one of these four megaspores survives. The functional megaspore is the starting point for the embryo sac development. The other three megaspores degenerate. The mitotic divisions. The nucleus of the functional megaspore undergoes three successive mitotic divisions. The nuclear division occurs without cell wall formation. The nucleus divides into two, and these two nuclei move to opposite poles of the megaspore. In the second division, each of these nuclei divides again, resulting in four nuclei, two at each pole. In the third division, each of the four nuclei divides once more, producing 8 nuclei. And these 8 nuclei undergo cellular differentiation and arrange themselves into specific cells within the developing embryo sac. The Allium type is named after the Allium genus, which includes onions and garlic. It is a bisporic type, meaning the embryo sac developed from the two megaspores. Here is the step-by-step breakdown. The process starts with the megaspore mother cell, that is MMC, within the ovule. It undergoes meiosis, resulting in 4 haploid megaspores. However, unlike monosporic type, here is what happens is after the first meiotic division, the two resulting cells, that is the dyad, remain connected. The second meiotic division occurs in each of these two cells, resulting in 4 megaspores, but they are still all held together. The two of these megaspores survive while the other two degenerate. Typically, it is the two megaspores that the chalazal end that persist. Each of these two surviving megaspores undergo two mitotic divisions. Nuclear division occurs without cell wall formation. This results in each megaspore containing 4 nuclei. The 8 nuclei, that is 4 from each megaspore, now organize themselves into cells within the developing embryo sac. The tetrasporic development stands out because unlike monosporic and bisporic types, all 4 megaspores resulting from the meiosis contribute to the formation of the embryo sac. This often leads to embryo sac with a large number of nuclei compared to other types. The Fritillaria type, named after the Fritillaria genus, is characterized by the presence of triploid nuclei. During the development, like all other types, the development starts with the megaspore mother cells in the ovule. It undergoes meiosis, producing 4 haploid megaspores that remain together in a tetrad. The 3 of the 4 megaspore nuclei migrate towards the chalazal end. These 3 nuclei fuse together to form a triploid nucleus. The remaining megaspore nucleus stays at the micropylar end and remains haploid. Now, both the haploid nucleus at the micropylar end and the triploid nucleus at the chalazal end undergoes mitotic divisions. Typically, each nucleus undergoes 2 mitotic divisions. The resulting nuclei organize themselves into the cells. The mature Fritillaria type of embryo sac has 7 cells, but the nuclei have varying ploidy levels. Egg cell and Synergids are haploid. Central cell contains a tetraploid secondary nucleus, and Antipodals have triploid nucleus. The embryo sac in the flowering plants is a vital structure for the sexual reproduction. Its components have specific roles to play in the process of fertilization and subsequent development of the embryo and endosperm. Here is a breakdown of the functions of each component. The egg cell is the female gamete. It is the cell that fuses with the one of the two sperm's nuclei from the pollen tube during the fertilization. This fusion results in the formation of a diploid zygote which will develop into the embryo. The two cells flank the egg cell are known as the synergids. They play a crucial role in attracting the pollen tube. They secrete chemical signals that guide the pollen tube toward the micropyle of the ovule. The pollen tube entry is facilitated by the synergids. The entry of the pollen tube into the embryo sac and the release of the sperms into the cells. They might also provide some nourishment to the egg cell before fertilization. The central cell is the largest cell in the embryo sac and contains two polar nuclei. The central cell plays a key role in the formation of the endosperm. The second sperm nucleus from the pollen tube fuses with the two polar nuclei in the central cell, resulting in the triploid cell. This cell develops into the endosperm, a nutritive tissue that nourishes the developing embryo. The antipodals are 3 in number. They are located at the chalazal end of the embryo sac. Their function is not clearly defined as the other components, but they are believed to have a role. They might provide nutrients to the developing embryo sac before fertilization. They usually degenerate after fertilization.